This is part C of the microbiology lecture for Unit 7. Specific immunity is the body's ability to recognize and defend itself against specific invaders and their products. Antigen literally means generates antibody. Antigens are the molecules that trigger a specific immune response. The epitopes are the antigenic determinants or specific 3D shape of the region on a molecule the body recognizes. The antibody also recognizes just a specific part of the antigen as well. The antigenicity of a molecule can be influenced by their size. Larger molecules tend to make better antigens. Molecules which are too small to be detected can become antigenic when bound to a larger carrier molecule called haptens. Haptens are often proteins. Exogenous antigens are outside of the cells of the body. These include microbes, toxins, secretions, components of cell wall membranes, flagella, pili, and some DNA released from dead bacteria. Endogenous antigens are inside the body's cells and hidden from the immune system. These can include microbes that are reproduced inside the host cell. The immune system can only respond to these antigens if they are incorporated into the cytoplasmic membrane. Autoantigens are antigenic molecules found on individuals' normal uninfected cells. These are normally eliminated during the development of the immune system. The lymphatic system is the home of many of our immune responses. The lymph is a colorless, watery liquid that arises from the interstitial fluid. The lymphatic vessels provide a one-way system to conduct the lymph. The lymph nodes often contain lymphocytes to attack foreign antigen and receive the lymph from numerous vessels. The red marrow contains stem cells that give rise to the cells of specific immunity. The lymphocytes are the smallest of the leukocytes and include B and T lymphocytes. They are characterized by large round nuclei with a thin rim of cytoplasm. The CD, or cluster of differentiation, are integral surface proteins found on cell surfaces. They are numbered in the order of discovery. The thymus is where T lymphocytes or thymocytes mature. It is located in the chest just above the heart. It is most active early in life and degenerates in adolescence. Lymph nodes consist of a medullary maze of passages that filter the lymph and house lymphocytes to mount an immune response against antigens. The cortex or outer portion has a tough cortex surrounding germinal centers where the cells of specific immunity replicate. The mucosa-associated lymphatic tissues include the other lymphoid tissue and organs such as the spleen, tonsils, appendix, Peyer's patches in the stomach, and lymphoid tissue in the respiratory tract. B cells arise in the red bone marrow in adults and mature there. They are identified by surface proteins. They are found primarily in the spleen, lymph nodes, red marrow, and Peyer's patches. Only a few circulate, but they all secrete antibody. Plasma cells are the clones of B cells actively fighting exogenous antigen. The antibody, or immunoglobulin, is the soluble proteinaceous antigen-binding molecule. This is sometimes referred to as a humoral response since the body fluids were called humors and is what is carrying these immune responses. And here we've got an illustration of an immunoglobulin. And you can see that we've got the different regions in here for attachment. Antibodies are composed of 4 peptide chains. The heavy chains are the 2 identical longer chains with the heavier molecular weight. The light chains are the 2 identical shorter chains with a lighter molecular weight. The light chains attach to a Y shape. The FC, or fragment crystallizable region, is the antibody stem. It is formed from the lower portion of the 2 heavy chains. These are named gamma, alpha, mu, epsilon, and delta for IgG, IgA, IgM, IgE, and IgD. The variable region is made from the arms of the heavy chain and light chain. The amino acid sequence of these varies from B cell to B cell, but will be the same for any given B cell. The 2 variable regions form the antigen binding site, known as the FAB, or fragment antigen binding. The FAB is complementary to the antigenic determinants. So here we've got the variable region in there. So when we say they're specific for any type of B cell, any B cell that's going to make one particular type of antibody, all of the antibody that is going to be made is going to be the same on those specific B cells and their clones. There are several functions associated with antibodies. One function is neutralization. It can bind to a critical portion of a toxin and prevent it from functioning in the body, or block the attachment molecules on the surface of bacteria and viruses. In opsonization, the antibody acts as a molecule to stimulate or enhance phagocytosis. Neutrophils and macrophages have receptors for the Fc region of IgG and will phagocytize the antibody and antigen it carries. Antibodies may activate complement and stimulate inflammation. With agglutination, several immunoglobulins bind to antigenic determinants at once and clump organisms together. This hinders pathogenic activity and increases the chance of phagocytosis. We have 5 classes of immunoglobulin. IgG is the most common, comprising 85% of the serum antibody. It is involved in complement activation, agglutination, opsonization, and neutralization. Its small size allows it to leave the blood vessels and bind pathogens before they enter the circulation. It is also small enough to cross the placenta, protecting the fetus. IgA is the secretory antibody, comprising about 5% of the total serum antibody. They exist as monomers connected with a J chain or joining chain to another polypeptide in various mucous membranes for a secretory component. It is present in tears, milk, saliva, sweat, and other secretions to produce the mucous membranes. IgM is the second most common antibody and is present during the initial stages of the immune response. It is 5 times larger than IgG because it forms a pentamer held together by a polypeptide J chain. This is more efficient because it has 10 antigen binding sites instead of 2. IgE is less than 1% of the serum antibody. It is not so important for agglutination, opsonization, or neutralization. Instead, it acts as a signal molecule for basophils and mast cells to trigger histamine release. It mediates between specific and nonspecific responses and is active with allergies and parasitic worms. IgD is not secreted by membrane-bound antigen receptors and is often seen in initial phases of the humoral response. Its complete importance is not known, and it is not present in all animals. It appears to play a role with B cell activation. IgD is just not nearly as well understood as the other ones, so we don't have as much information on that one. The B cell receptor, or BCR, on an antibody is going to be a receptor that remains integral to the cytoplasmic membrane. Each B lymphocyte has 250 to 500,000 identical copies and has 2 antigen binding sites like other antibody. Each is randomly generated, so there's 10 to the 11th different B lymphocytes per person. And so that seems like a really big number, but there are lots of different things that can be a potential antigen. And so we need to have lots of different B cell receptors that are going to be able to attach to those different antigens in there. If you were to make one of these that attacks your own tissues, those are the ones that you want to have destroyed during your immune system development to avoid having an autoimmune condition. T cells are lymphocytes that leave the marrow and mature in the thymus. The TCR, or T cell receptor, is composed of 2 different polypeptide chains with a groove that acts as an antigen binding site. The TCR binds complementary antigenic determinants. There are at least 10 to the 11th different possible sites. In the CMI, or cell-mediated immunity, the T cell acts directly against the antigen and does not secrete antibody. CMI, or cell-mediated immunity, is involved with intracellular invaders. Each TC has hundreds of copies of unique TCRs and the presence of a Cd8 positive marker. They directly kill cells infected with intracellular pathogens and abnormal cells. The TH, or T helper cells, are Cd4 positive, meaning they have the Cd4, or cluster of differentiation 4, marker on them. They help regulate the activity of B cells and T cells and secrete various cytokines which determine which immune response will be activated. The Th1 subset assists with the TC with the infection response. They are CD6 positive and have cytokine receptor 5, or CCR5. The Th2 subset functions with B cells and in allergy and hypersensitivity responses. They have CCR3 and CCR4. The cytokines are soluble regulatory proteins that act as intercellular signals when released by certain cells. The ILs, or interleukins, signal among leukocytes. The interferons are antiviral proteins which may also act as cytokines, such as gamma interferon. Growth factors are proteins that stimulate cell division to ensure the presence of adequate leukocytes. TNF, or tumor necrosis factor, is secreted by T cells and macrophages to kill tumor cells. It also regulates the immune system and inflammation. The cytokines signal the leukocytes to go to the site of inflammation and activate other leukocytes. These are a big part of the regulation of the immune system. Clonal deletion is the important process where the body eliminates cells which are self-reactive, since the receptors are randomly generated. Apoptosis will occur when a lymphocyte binds an autoantigen. The HLA, or human lymphocyte antigen, is a set of glycoproteins on the cell membrane of most cells. These were named HLAs because they were first identified on human leukocytes. The MHC, or major histocompatibility complex, is coded for by a set of genes on chromosome 6. The class 1 MHC, or MHC1, is found on all cells, including red blood cells. The class 2 MHC, or MHC2, is found on B cells and antigen-presenting cells, or APCs. The APC, or antigen-presenting cells, are monocytes, macrophages, and their relatives that are infected cells presenting an antigen. The HLAs are important with a lot of autoimmune conditions in there. They're markers that can be looked to see if a person potentially has that condition, because they will be present in most cases of those diseases. The size of the antigen may affect the immune response. A T-dependent antigen is larger with repeating antigenic determinants. B cells can bind directly to large antigens, with the repeating subunits cross-linking numerous B cell receptors or BCRs, essentially providing its own co-stimulator. This stimulates proliferation to make plasma cells and antibody without the help of T cells. Most antigens are T-dependent. They are smaller with less accessible antigenic determinants. The B cells targeted cannot function without the help of the T helper cells. Humoral immunity is the response against exogenous pathogens. T-dependent exogenous antigens require T helper cell assistance. B cell activation and clonal deletion begins with the antigen presenting cell presenting antigenic determinants to the T helper cell and activates the T helper cell with the complementary binding site. The T helper cells are Cd4 positive and have unique TCRs. The Cd4 must bind MHC2. The Tc signals the Th cell to differentiate with IL-1 to become Th2. In clonal selection, only the complementary B cell binds the Th2 and IL-4 activates the B cells to proliferate and produce memory cells and plasma cells. So the Cd4 and Cd8 markers are really important because they have to bind with the MHC as part of the immune response. The way I remember them is they always have to add up to 8. If you were to multiply MHC1 times the CD8, they come out to be 8. If you multiply CD4 with MHC2, 2 times 4 comes out to be 8. Plasma cells are large activated B cells. They only produce antibody identical to the parent cell BCR, which results in the production of thousands of antibodies. These secondary cells are short-lived, lasting only a few days. The antibody can remain in the body for months, and some clones can persist for years. The memory B cells do not secrete antibody, but survive with the BCRs complementary to that specific antigen. Memory B cells can survive for years in the lymphoid tissue. During a primary response, a relatively small amount of antibody is produced. It may take days or weeks to eliminate the antigen or toxoid from the body. During the secondary immune response, memory B cells make a reserve of antigen-sensitive cells that activate with another exposure to that toxin or antigen. The cells proliferate and differentiate rapidly without interaction with an APC. Large amounts of antibody are present within a few days, giving a faster and more effective immune response. The memory response is enhanced with subsequent exposures to the antigen. So when you've got the subsequent exposures and the memory cells in there, you're going to get a stronger, faster response, which is the goal of vaccination in there, to help prepare your body so that it can give a larger, faster response when you're actually exposed to the actual antigen. The other thing is it does not need a co-stimulator anymore. It's already been co-stimulated in a sense that it just needs to find the antigen and it's good to go. In the cell-mediated immune response, the body must fight intracellular pathogens in abnormal cells. When the cell synthesizes protein, it displays a fragment of the protein, including viral proteins, in the antigen binding groove on the MHC1. Certain APCs secrete IL-12 to activate the T helper cells to become Th1s. The Th1 cells secrete IL-2 and gamma interferon to activate the TC with the TCR complementary to the antigen. The activated TC produces memory T cells and more TC that produce IL-2 and IL-2 receptors to become self-stimulating. At this point, they no longer need the APC or TH. The activated TC binds infected cells with its TCR and with CD8 to make its MHC1. The target cell is killed through the perforin granzyme path or the CD95 path. Perforins are a cytotoxin which aggregates into a tubular structure on the infected cell membrane. Granzymes move through the perforin channel into the target cell and activate apoptosis. The TC moves on to another infected cell. The CD95 is present on most cells. The TC cells have CD95L, a receptor. When the CD95L, which stands for ligand, binds CD95 on the target, it activates enzymes for apoptosis. Memory T cells are made from activated T cells. They can persist for months to years in the lymphoid tissue, and subsequent exposures to antigen matching the TCR responds immediately without an antigen-presenting cell to produce the TC. This response is more efficient. There is regulation initially to prevent responding to an autoantigen because co-stimulation is required from an APC.